better understand resistance mechanisms, which are still poorly understood in Aedes albopictus. And we have to detect this resistance early in the field. So here you have the major mean of insecticide resistance. So you have comportamental resistance, which consists in avoiding the insecticide by adaptive comportamental things. You have cuticular resistance, which consists in modification or in thickness or in composition of the cuticle, so that the cuticle is less permeable to insecticide. And you have also target resistance by target modification, which consists in a conformal modification of the target of the insecticide, which impairs the binding of the insecticide to its target. And the last one is metabolic resistance, which consists in an improved activity of detoxification enzyme. And those detoxification enzyme can either sequester the insecticide or degrade it. So there are a lot of different resistance mechanisms, and they often occur in natural population. That's why studying them is not often easy. So to be a bit more precise about resistance by target modification, usually the target of chemical insecticides are neuronal proteins. So they can be, they can be voltage dependent channels. They can be neurotransmitters or receptors or neurotransmitters metabolizing enzymes. And in the case of pyréthroïdes, the target is the voltage-gated sodium channel. And the associated mutation are called CADR mutation or knockdown resistance mutation. So given the fact that CADR mutations are punctual mutations, they are easy to characterize, and diagnostic assays are often available to track them in the field. About metabolic resistance, as I just described, the insecticide can be sequestered or directly excreted, or it can, it exists different phases during which the insecticide is during phase 1, the insecticide is the insecticide is make more hydrophilic to be excreted more easily. And if it's not sufficient, it is conjugate with hydrophilic component to be excreted later. So the main enzyme involved during phase 0 and during excretion are ABC transporters. During phase 1, the main enzyme are P450, and during phase 2, the main enzyme involved are GST and UDP-GT. So what are the genetic mechanisms underlying this increased activity of detoxification enzyme? You can have a punctual mutation in the active site of the enzyme. which make it more efficient, or you can overexpress detoxification enzyme, either by a modification in copy number of the gene, or a modification of regulation of gene expression, either by a remote element in the genome, which we call transregulation, or modification directly in the promoter region of the genes, which is called cis-regulation. So characterize metabolic resistance is a bit hard because there are hundreds of genes potentially involved. There is functional redundancy. That is to say, one insecticide can be detoxified by a different enzyme, and one enzyme can detoxify several insecticides. And there are a lot of different genetic mechanisms. So usually there are less diagnostic assays available. So my work is about identifying the genetic determinant involved in deltamethrin resistance in albopictus. And for that, I study 5 regions. I study Africa with the Central African Republic and Gabon, Italy, metropolitan France, Asia with population from Laos and Cambodia, and La Réunion Island. And for that, I used high throughput sequencing approaches, and I combined transcriptomic and genomic data to find out deltamethrin resistance marker in this mosquito. So what sample so the first step of this work was to collect 6 to 7 find population in each different region, and then we pull them together to create one composite population by region. Then we split this composite population into 2 populations, one maintained in the laboratory without insecticide treatment, and one selected with deltamethrin at each generation, with a selection strength of about 50% mortality. And the sample I use for my genomic and transcriptomic and genomic analysis are here. I have 2 susceptible reference lines, which are SPAM originating from the south of France and SREN originating from La Réunion Island. And for each regions, I have samples from the non-selected line and the selected line. And for La Réunion Island, I have also free find population. So here you can see that the number of generation of selection are different for each population because we didn't resave them at the same time. So to identify those resistance markers, we have to compare samples together. And I used the same comparisons for transcriptomic and genomic analysis. So the experimental design is a bit different for La Réunion Island because we have find more population. But basically, comparing the selected line versus the non-selected line allows us to find out genes linked with deltamethrin resistance and selected in the laboratory. But we are susceptible to have false positive due to genetic drift or bottleneck effect. So to reduce the number of false positive, we use the cell versus susceptible reference line comparisons, and we retain only the genes which are linked to the delta resistance in the pre-comparisons. And for La Réunion Island, it's the same thing, but we use only SRN as a susceptible reference line because it's originating from La Réunion. And we use also the free filed population, which we compare to SRN. And when we look at the genes present in all those three comparisons and linked with delta methylene resistance, it gives us genes selected in the file in La Réunion. So for La Réunion Island, at the end, we have the genes selected in the file and in the laboratory. Then we assigned the genes to, or not, to a candidate gene category. Those 12 categories correspond to families known to be involved in insecticide resistance. And we assigned 953 genes to those categories in albopictus. So there are a lot of detoxification enzyme and cuticular proteins. So about the bioinformatic analysis, for transcriptomic, for each sample, we use 4 replicates of 25 females that we sequence with the DNBSEC technology with reads of 100 base pair in per end. Then we filter those reads on base quality and remove adapters. And then we map those reads on the AALBF3 transcriptome. And we use those reads for 2 things. To identify alternative splicing events and indels. Then we map those events on the transcriptome, and this allows us to find alternative splicing events, indel and intron retentions, which are linked with delta methylene resistance. And we do that in collaboration with a team of the LBBE of Lyon. Then for genomic analysis, we use sample of 100 females that we sequence with the DNBSEC technology in 150 per base read in per N. We filter all the reads and map them onto the genome, and then we filter reads on mapping quality and remove duplicate. And once obtained those filtered reads, we use them to study different things. The first thing was to calculate the ratio of standardized exonic coverage to find out variation in copy number of genes linked with delta methylene resistance. We also work with a team of ISEM on a transposable element linked with delta methylene resistance. And why I'm here in MIVEGEC is to study polymorphisms associated with delta methylene resistance. So the first thing that we do was to characterize the delta methylene resistance phenotype of composite population. To do that, we exposed 3 days old female to 0.03% delta methylene papers during 1 hour. And Yes, for the rest of the presentation, I will present result for La Réunion separately because the experimental design is a bit different. But here, what we can notice is that between the non-selected line and the selected line, in all the studied region, we have a decrease in mortality after several generations of selection, which means that resistance alleles are probably circulating in all those countries because we can select them. In La Réunion Island, the resistance have been detected since 2017, and here you can see that the non-selected line is more resistant than the susceptible reference line, and that the free-fight population have more or less the same resistance phenotype, which is expected. And the selected line is very resistant after only 5 generations of selection. So the first thing we look at in our genomic data was cardiac mutation. The problem was in our version of the genome. The gene of the voltage-gated sodium channel was not annotated, so we manually annotated it. And then we searched for non-cardiac mutation in Aedes mosquitoes. And we detect nothing in La Réunion, in Africa, and in Asia. But in Europe, in Italy and France, we detect the V1016 gene mutation and the I1532T mutation. And the percentage here are not representative of the frequency in fight population, but the absence of those mutation in the non-selected line tells us that the frequency is probably very low in fight population, but present because we can select it. And in France, this mutation was present in the non-selected line at a high frequency. And so we find those 2 mutations both in France and Italy populations. About my transcriptomic results, among all genes detected, we identify 734 genes that were candidate gene and belong to family known to be involved in insecticide resistance. And here you have a summary of over-transcribed and under-transcribed genes detected, and between parentheses, the candidate genes. And what we can see here is that there are few genes in common between each regions, and only 1 candidate gene in common between France and Italy. So we can say that resistance mechanisms are probably well differentiated between regions. Here, I present only candidate genes linked with delta-methrin resistance. And what you have here is a heat map. So here you have the log2 fold change of the difference of transcription level between the cell line and the susceptible line here. So, for example, this gene is probably 8 times more expressed in the spam line is 8 times more expressed in the selected line as compared to the spam line. The circles here indicates when the gene is significantly overexpressed, and when the cycles are full, it indicates that there is a significant variation in gene copy number. So in Africa, there are a lot of cuticular protein overexpressed, and P450 belonging to the CYP6 family, which is known to be involved in pyréthroïde resistance in mosquitoes. In France and Italy, the candidate genes belong to different candidate families. And we have one CYP6 gene in common between the two regions. And this is probably not a false positive because it's present in two geographically close regions. And in Italy, we have GST, which is overexpressed and with a copy number variation. And in Laos, we also have a gene overexpressed with a copy number variation, which is this esterase. So if we zoom again, here you have the gene overexpressed, and the genes overexpressed and the gene with copy number variations. And in Italy, this candidate gene, which is the GST data, we can see that it's potentially duplicated in the selected line. Here is the genomic coverage in red of the selected line. And the overexpressed esterase in Laos is here and seems to belong to a big cluster of esterase, which is duplicated in the selected line. For La Réunion Island, all the candidate gene belongs to the chromosome 1 and chromosome 2. There are 3 chromosomes in Aedes albopictus. And some of them are in cluster, like those UDP-GT, those CYP6, and those 2 ABC transporters. And we can notice that all the gene present here belongs to the families that are mainly involved in metabolic resistance, so they are strong candidates. And in black, it's when ortholog genes in another mosquito have already been proved to metabolize pyréthroïde insecticides and are subfamily which are known to be involved in pyréthroïde resistance. So they are very strong candidates. Okay. In La Réunion, there is one CYP6 gene which is overexpressed and have a significant copy number variation, an augmentation in copy number variation in a resistant line. But the structure of this duplication is not very clear. The duplication is bordered by repeated elements, and it seems to have 2 haplotypes of this duplication in our line. But the presence of this duplication still have to be confined. And in La Réunion Island, when we found, when we look at alternative splicing data, we found in indel of 6 base pair in transcription factor HR96, and this transcription factor expresses a variant protein in the resistant line. And this transcription factor is known to upregulate P450 in another insect. So it's potentially also a good history. So to summarize, we found resistance in the field in La Réunion and also in metropolitan France. We found a copy number variation in resistant line for gene overexpressed, so 1 P450 in La Réunion, 1 GST in Italy, and 1 esterase in the population from Laos and Cambodia. In France and Italy, we find out 2 cardinal mutations that are circulating in 5 population. And we detect 1 P450 common to Italy and France. So yes, in La Réunion Island and in Asia, the gene circulating in 5 population are linked with metabolic resistance. In Africa, we have a lot of cuticular gene involved in cuticular resistance and metabolic resistance. And for Italy and France, we have both target modification and metabolic resistance. And so this work is still in progress. And for now, we are searching polymorphisms associated with resistance. So new genome of albopictus has just been not yet published, yes, but we use it to map our roots onto the new genome. And then we plan to do genome scan analysis and diversity measures to find those polymorphisms. The next step is then make a link between all the different type of information that we have. And the long-term objectives of my work is to identify in each different regions which are the best DNA resistance markers, to design diagnostic tools to detect them in the natural population, to better predict the risk of resistance and manage it. So thank you for your attention. And if you have any question, I can answer it. But it's a lot of information. Yes. Yes.
Est-ce que tu peux parler dans le
You
micro
can
pour les gens en Zoom?
ask your question in French if you prefer.
I don't know. Should okay, I will try in English. Let's stay in English. You show few SNPs, a single nucleotide polymorphism selected, and you said one was at 0%, so was kind of non-existent. You just mentioned that it's probably there because we succeeded to select this mutation.
Yeah.
But this is a term probably there. So did you check that it was there, or it's just an assumption? Is 0%, but maybe you have seen that, yes, it's at least present one times, but it represents less.
We have my samples is 100 females.
Yeah.
And I have a genomic coverage of about 100 So 1% is about one mosquito. So probably there is one mosquito in my.
Yeah, so
Yeah,
it's
right.
probably you didn't check really if the mutation is there.
The non-selected line and selected line are from the same population that we split in two. So if we can select it, it's probably here, but we can lose it by, I don't know, genetic drift or a bottleneck effect, probably.
Yeah. Okay.
I don't know. But yes, so those two mutations have already been detected in other studies.
We agree that what you align here is the genome. Yes, genomic data. The reads is not transcriptomic. Here is genomic.
Genomic data.
Yeah, yeah. Okay. Thank you.
Yeah, so when you check this mutation is when you collect them or after you're doing your breeding? Is it like is there J6 is the sixth generation when you check if they have the mutation? Et si quand tu as regardé là pour la mutation, c'est à la génération 6 ou tu as regardé quand tu as récupéré les populations sur le terrain?
Oui, en fait, oui, la population française a été sélectionnée que pendant 6 générations. Donc ça, c'est les pourcentages après 6 générations de sélection. C'est pour ça que tu ne peux pas vraiment comparer et de toute façon, ces pourcentages-là ne sont pas représentatifs de ce qui se passe vraiment dans les populations naturelles.
Et du,
Ça permet de dire que c'est présent ou pas.
et du coup, vous n'avez pas le T0 de savoir qu'est-ce que c'était à l'origine avant de sélectionner ou pas sélectionner vos populations? De savoir avoir.
Sur la population composite quand on
Ouais.
Non, mais on peut se dire que bon, vu que la lignée non sélectionnée n'a pas été sélectionnée, ça peut être un proxy de ce qu'il y avait à l'origine, mais bon, il peut y avoir une.
Des questions, Zoom? D'autres questions?
I just wanted to add some je peux le faire en français, information à propos de ma question. En fait, elle était liée à une idée qu'il y avait derrière. C'était est-ce que ça pourrait pas être lié à une enzyme comme l'enzyme ApoBec connue dans d'autres organismes qui aurait modifié l'information au niveau génétique? En fait, c'est pas la mutation est déjà présente dans un des moustiques et puis qui va partager sa mutation, mais plutôt un élément extérieur comme l'ApoBec qui ferait la mutation au niveau donc génétique et puis après elle est transmise.
La mutation serait apparue pendant les 6 ou 13
Dû à la fait de la sélection, finalement, tu actives une enzyme qui n'est pas censée être très active là, à ce moment-là. ApoBec est connu, mais peut-être qu'il y en a d'autres. Je ne suis pas trop dans. C'était juste dans cette idée-là, est-ce que ça a été pensé ou pas, ce potentiel? C'est pour ça que je voulais savoir, est-ce que tu l'as vu au moins une fois vraiment pour savoir si ça vient extérieurement ou si c'est juste.
La
transmis.
mutation, oui, on la voit là.
Oui, là, mais dans le 0%, est-ce que tu allais voir si elle était présente au moins une fois?
Oui, j'ai regardé.
Ok.
On a rien. Mais bon, c'est peu probable parce qu'on trouve quand même ces mutations, on sait qu'elles sont.
Ouais, tu me disais, oui, je comprends. Mais c'était pour donner l'idée pourquoi je posais cette question. J'avais une idée derrière la tête.
C'est ça.
Des questions? Bon, on va arrêter là, du coup. Merci beaucoup, Tiphaine.
Merci d'avoir.